everybody. Welcome to the next latest installment of the Research Introductions. My name is Gary Radice. I'm in the biology department here, and it's going to be fun for me to tell you about the research that's going on in my lab this evening. What I study is the development of amphibian lymphatic hearts, and I'm going to guess that probably not too many of you know what a lymphatic heart is. So I'm going to start there, and then tell you why they're the most interesting thing in the world to study. So, I've been working on different aspects of frog development ever since graduate school, and this is the frog that I work on. This is Zebus Leavis. It's the South African clawed frog. It's, as the name implies, it's native to South Africa. And but we have the ones that we use live upstairs on the third floor, and we refer to them as amphibian Americans. So they're unique for several reasons. One is, as this picture shows, you see it living in an aquarium. So most frogs that you're familiar with, you probably think of them as sitting on a lily pad or being out not underwater all the time. But this frog lives its entire life in the water, so you can raise them like fish, which makes them really easy to raise in the laboratory. They also will eat fish food, just pelleted fish food, so you don't have to feed them live flies, which is really nice for taking care of frogs. So they're very easy to raise in the laboratory. They have a couple of interesting, the adults have a couple of interesting features. One, you can't see very well here, but they have claws on their feet. Most frogs don't have claws. And for our purposes, the real reason that we use them is that it's easy to get embryos from these year-round. So you can simply inject them with a hormone, and the next day they're shedding eggs. And so that's, and most frogs you can't do that with. So they're wonderful laboratory animals. What I'm interested in, so people use them to study various aspects of development. What I'm interested in is studying the lymphatic hearts. So I have to tell you a little bit about the lymphatic system first. Everybody knows about the blood circulatory system. You have a heart that pumps blood through arteries and arterioles, through capillaries, and then into veins, and then back to the heart. And for the blood to continue to circulate, it needs a heart, some sort of muscular pump to move that. So all animals that have, almost all animals that have blood also have a heart to pump the blood. Now, most animals also have, most vertebrate animals, also have another set of vessels that move a different kind of fluid, and these are the vessels that move lymphatic fluid. Most people aren't really aware of their lymphatic system because in mammals, you and me, we don't have any hearts that pump lymphatic fluid. So there's no pulse that you can feel, there's no place on your body where you can say, oh yeah, I can feel the lymphatic fluid moving, because the lymph system in all animals is not circulatory. There is no equivalent of an artery or a vein in the lymphatic system. It's all one-way vessels. So they start out here in the peripheral tissues, out in the tips of your fingers or out in the periphery of your body as dead ends. And fluid from the body's tissues leaks into the vessels, like around in here, and then begins to flow backwards into larger and larger vessels. And then finally in mammals, and you and me, all that lymphatic fluid eventually empties into your bloodstream and near the heart, and it's returned to the heart, and then it circulates that way. So it goes from, so the lymphatic fluid goes from your blood, squeezes out of the capillaries into your body tissues, and then the excess fluid is picked up by the lymphatic system and carried back to your heart. What moves the lymph in you and me is simply the normal everyday movements of your muscles. It massages those vessels, and as I'm walking around like this, my muscles are contracting and relaxing, and that's sort of squeezing these vessels, and eventually that pushes the lymph back to the bloodstream. You can realize this when you wake up in the morning. If you haven't been moving for a while, if you've been sleeping all night, you notice your face is maybe a little bit puffy, and that's because the lymphatic fluid has accumulated in your head while you've been lying there. And then once you get up, gravity will help drain it, and your movements will help drain it, and so on. Or if you stand in one place for a long time, and your feet begin to swell, that's because you're not moving, and the lymphatic fluid is beginning to pool in your lymph vessels in your legs. Frogs have a special problem with moving lymphatic fluid around because their skin is permeable to water, and their tissues are salty, and they're sitting in fresh water. So they're sitting in an osmotic situation where pond water is flowing in down its osmotic gradient into their body through their skin. So in addition to the fluid that leaks out of their bloodstream into their body tissues, they also have to worry about water that's coming in from the outside world into their body tissues. So they have a lot of lymph to move around, and frogs are not all that active. Being cold-blooded animals, most of the time they're just sitting there. So they have a problem in moving lymphatic fluid around, and one of the ways that that's been solved in evolutionary terms is that they have tiny hearts, little booster pumps. They keep the lymph moving. So here is a diagram made back in the late 1800s of a bullfrog tadpole, and here is the lymphatic system's outlined here. What they did was they took some India ink and simply injected into the lymphatic system, and it outlines the vessels really nicely, and then they drew this nice little picture showing you a pair of lymphatic hearts, one here and one here. Later on in development, as we'll see, this frog will develop another 3 more pairs back here, right in here, 1, 2, and 3 back here. You can't see them yet. It's not old enough to develop back here. All right, so amphibians develop these lymphatic hearts during the tadpole stages, and then those hearts persist into the adult stages, and I'll show you that in a little while. So let me show you a couple more pictures. Okay. So this is a drawing from the 1800s. These are some pictures taken in my lab a couple of years ago in which the lymphatic hearts, muscles in the lymphatic hearts were stained with an antibody against a muscle protein. And so here you can see the axis of the back of the frog, and here's a lymphatic heart over here, and here's one over here, and here's one enlarged a little bit so you can see it. So these are the lymphatic hearts, and it's the ones in the front of the tadpole are the ones that I'm going to be telling you about today. We know a little bit less about the ones in the back of the tadpole. One of the reasons that I really like this project is that I didn't know about these lymphatic hearts until a student in one of my developmental biology classes pointed them out to me about 8 or 9 years ago. And we had some tadpoles from the frog that I showed you anesthetized, lying in some dishes, and we were looking at them, looking at their anatomy. The tadpole that I work with happens to have transparent skin, so they're really nice for doing these kinds of studies. You can anesthetize them, they sit in a dish, and you can look in, you can see all the organs right through the skin. And one of my students was looking at it through a stereo microscope, and she said, "Dr. Radice, what's this thing beating?" And I said, "A heart." Trying to be nice. And she said, no, there are 2 of them, and they're on its back. And I said, "What?" And so I sort of pushed her aside and said, "Let me see that." And sure enough, it looks just like this. And I'll show you some pictures in a minute. I'd never heard of them before. I'd been working on frogs for 15 years. And so I decided I needed to do a little reading about this, and it turns out that they turned out to be really interesting structures to study. All right, so here's a tadpole from the frog that I work on, from Zebus, that I took today, actually. It's lying in a dish, and just to get you oriented a little bit, here's its left eye, and let's see, this is its brain. Okay, so you can actually see the brain right through the skin. It's got transparent skin. This is the gut back here. This structure right here is an embryonic lung. Okay. This big structure right here is going to be the frog's ear. It's called the otic vesicle. It's going to be his ear. So just to get you oriented here, the head of this tadpole ends about here, and then this is all the body of the tadpole, and then you can see the tail going off this way. The lymphatic hearts are really small. So you can't see it in this picture because of the way the lighting is, but it's really small, and it's going to be right about here. They're about 1/10 of a millimeter long. Okay, so a millimeter is a 25th of an inch. These are 1/250th of an inch. So you can't see them without a microscope, without at least a stereo microscope. You have to magnify them a little bit, or you can't see them. Okay? Which is why most people miss them unless you're actually looking for them. But they're very small, and they're transparent. So they're pretty hard to see. I'm going to show you a movie of them in a a minute. If they're beating, they're relatively easy to see. All right, so, okay. So this is an enlargement of the animal you just saw. Here's that lung that I showed you. This right here is the embryonic front limb. So we're right up here on you and me. This would be the equivalent of being sort of in the shoulder blade region. Okay. This is a nerve. It's running out from up here in the spinal cord, and it's running back this way. And this structure right here is one of the lymphatic hearts. And let me show that to you. So if this works, if you're lucky, you'll be able to see it. So there it is, twitching away, and it should loop so you can see it again. It's a large picture, and it's sort of hanging up on my laptop here. But that's the lymphatic heart right there. So this isn't the best movie in the world. It should be beating around once a second, about the same as your heart. Okay, so and the hearts on both sides beat at about the same time. Okay, so these are lymphatic hearts. What do we want to know about them? Well, the first thing that I was interested in was what's the structure of these lymphatic hearts? How are they actually built in 3 dimensions? Turns out there's not a whole lot of information about that in the literature. There's some, and there's some pictures of individual cells, but the actual 3-dimensional structure, how all the cells are arranged, how many of them there are, that wasn't known very well. So one of the projects that's going on in my lab is to try to get a better idea of what the three-dimensional structure of these hearts looks like. Okay, so the first thing that we did was cut some histological sections. So we took some tadpoles and we fixed them and embedded them in wax and cut sections through them and made slides, stained the slides, and this is what it looks like. So here's that image that I showed you before. That's a whole tadpole, and here's a section through one of those lymphatic hearts. Here's the muscles along the back. That's these muscles right here. This structure over here is the limb coming out, the forelimb that you saw, that I showed you before. And here's one of the lymphatic hearts right here. It doesn't look like much. It's a hole. So that's the space inside for the lymph. But what I'm interested in are the structures that are around, the cells that are around it. Here's an enlarged view of a different lymphatic heart with a little better resolution. So here's the space for the lymph. This is part of one of the valves. There's a little valve in there to keep the lymph flowing in one direction. So the lymph comes in, squeezes the lymph out. And there's a valve that shuts back here so the lymph can't flow backwards. That's part of that valve right there. And these little stripes right through here, like there and like there, and you can see a little bit up here and here. Those are the muscles that power that heart. And so most of what I'm going to be telling you about is about those muscles. That's for right now, that's the most interesting thing for me. I want to know what kind of muscles those are, where they come from in the embryo, and how do they relate to the other muscles in the heart, other muscles in the embryo. So that was sort of a low magnification view of the lymphatic heart. This is a higher magnification view with the transmission electron microscope, showing you what some of these muscle cells look like. If you've had cell biology yet, you might have seen some pictures of skeletal muscle before, or heart muscle. And these are some of the structures inside one muscle cell that actually cause the heart to contract. So this structure is called a sarcomere, and what it does in any striated muscle cell is that this is the unit that contracts. There are filaments here, and they slide back and forth like this whenever the muscle contracts. So these are genuine muscle cells, and they look a lot like the cells in the blood heart. They don't look very much like the cells, like skeletal muscle cells, and that's that goes along with their physiology as well, the way that they beat. So these initial studies say that these lymphatic heart muscles are a type of heart muscle. It's not too surprising, but it is good to confirm that. Okay. Here's that tadpole again. Remember I said that there'll be another set of lymphatic hearts, 3 or 4 sets, that develop posteriorly, and they persist into the adult. This is actually part of an adult frog, and I've peeled back the skin to show you this structure right here. What you're looking at is, here's the left leg of the frog going out this way, and here's the right leg of the frog going out that way. So this is right at the base of the spine, right on its back. It's right in here. And there are actually 4 lymphatic hearts. Doing the dissection causes some of the blood to leak, and it ends up in the lymphatic fluid. So you can see where the hearts are because there's blood in this preparation. So there's 1, 2, 3, 4, and 1, 2, 3 maybe, and 4. Okay? So you can see these lymphatic hearts beating in the adult as well. And if you can put it under a microscope, and here are 2 of these lymphatic hearts right here, one here and one here. And you can see them twitching away, embedded in this little pad of fat. Right? So even in the adults, this animal has these lymphatic hearts. Okay? So amphibians have these, reptiles have them, and some birds have them, but not mammals and not fish. So to find out what these look like in 3 dimensions, we're trying several different things. Here's that one you've already seen before. And what we did in this case was we wanted to get a higher resolution view in 3 dimensions. And so what we did was prepared some of these lymphatic hearts for the scanning electron microscope. Now what this instrument is mostly for is for seeing the surfaces of objects. What I want to use it for is to see both inside and outside. So it's a hard task for the scanning electron microscope to do. But in this preparation, what we did was just peeled off the skin on the top of a tadpole, on the surface of a tadpole, and then we could have a view of the lymphatic heart inside. So this is one of these lymphatic hearts, and these are those muscle cells. So here's one right here. So notice that this cell is branched. So you can see it branching like this. That's a feature of cardiac muscle, blood heart muscle. That's something you don't see in skeletal muscle. So this is some additional information that one of these is, that this is really is a type of a heart. Now what I'd really like to do is, this sort of tells you in general what it's going to look like. We have this open meshwork, this sort of basket work of these individual heart muscle cells. But it's a little hard to count them. You can't see what's on the other side. You can't see what's inside the heart very well. So another approach that we're using to try to figure out the 3-dimensional structure is to use the Confocal Microscope, which uses a laser to look inside the heart. So what you would have, to do this experiment, you have to make the cells fluorescent first, which is a trick, and then use the laser to look inside. And then using that microscope, you can optically section the heart. It's sort of like an MRI, but it's in color. And so we're just starting. I just started to do some of that, and this is a reconstruction, sort of a computer image of what that microscope saw. In this case, we didn't actually see the whole cells. That was a little too confusing, but what we did see were the nuclei. So everything in purple here is the nucleus of a cell in a lymphatic heart. And these are the cells that are in the lymphatic heart, and the blue ones are the cells that are around the lymphatic heart. So one of the nice things about the confocal microscope is that it's all really done by a computer. The cameras take the picture, and the computer can redraw those pictures any way you want. So you can make 3-dimensional reconstructions like this, and it's a lot more obvious what's happening if you can rotate the image. So you'll notice as soon as the image starts rotating, you can see it's 3 dimensions and not just a flat object. So we're on our way with this one. You'll notice that it's in this picture, though, it's really only half of a heart. So we've got half of it. We don't have the other half. It's actually a little bit too thick to image clearly in this with the confocal microscope. So we're trying to figure out some other improvements, modifications to this to get the whole thing in the picture. Okay, so one of the projects is to understand the 3-dimensional structure and of the heart, and also the types of muscles that are in that heart. So that's sort of a structure problem, but more interesting to me, or another interesting problem, is where the lymphatic heart muscles come from in the embryo. This is the real developmental biology problem. First one was kind of an anatomy problem. This is a question about embryonic development. All right, so why is this an interesting problem? Here's the story. We know that if you take these pictures like this at different stages of development, if you go early enough, like maybe 3 days of development, there's no lymphatic heart there. What you see is a lymphatic vessel, but there's no muscles on it. It's just a chamber of a vessel. And then between day 3 and day 4, what happens is cells begin to show up. These are little cells like this. They begin to show up, migrating through the embryo, and they glom onto the surface of that vessel, and then differentiate into these muscle cells. So the muscle cells for those hearts come from someplace else. And what I want to know is where they come from. So if you can imagine this, you've got a little vessel there, and in another part of the embryo, there are some cells that are specified to be lymphatic heart muscle cells, and they just crawl through the embryo until they find this place, and then they form the heart. So that's really interesting. And how does the embryo do that? How do they know where to go? How do they know to become lymphatic heart muscle cells? Those are the questions that I'm interested in. Okay, so we have some clues as to where they might come from. And these are some cartoons of a chicken embryo. I don't have good equivalent ones from a frog's, but the story is fairly similar. To get you oriented here, what you need to know is what everybody thinks they know about where muscle cells, all the muscle cells, come from in a frog, or a chicken or a mammal. They all are thought to come from the same place, and they're thought to come from this tissue right here, which develops as a set of blocks. There's one here, and one here, and one here, and one here, all the way down the side of the animal, just under the skin. This is the skin, and right next to the spinal cord. So this is the spinal cord. This is a rod of tissue just outside the spinal cord and just underneath the skin. Some of the cells in here, right here, are going to form muscle cells, and all of the skeletal muscle, that is, all the muscles in your back, all the muscles in your limbs, all the muscles in your belly, they all come from this patch of tissue right here, which then migrates away and forms the skeletal muscle everywhere else. The lymphatic heart in a frog forms right here, right at the bottom edge of this mass of tissue. So it forms heart tissue right next to a region that forms skeletal muscle tissue. Okay, so here's another cartoon view of it in 2 dimensions. It's these cells right here that are going to migrate away and form, let's say, the cells in the limb. The lymphatic heart, if this was a frog, would form right about in here. So that lymphatic heart is forming in an area where muscle cells, skeletal muscle cells, are migrating by. Okay. Let me summarize that here. This is a frog embryo. And to get you oriented, here's the spinal cord right here. Here's that mass of tissue that we saw red in a chicken right here. That's what it is in a frog. And that's these cells right here, or right here. And the lymphatic heart hasn't formed yet in this embryo, but when it forms, it's going to form right about in here. So it's forming these heart muscle-like cells in an area right next to it, a region that forms skeletal muscle cells. So I think that somewhere in this mass of cells that form skeletal muscle, there are a few that become specified to form lymphatic heart muscle. And so I think that's true, but I haven't been able to prove it yet. So that's the main project that I'm working on right now. All right, so let me tell you, I've tried several different things. Let me show you one of the approaches. If you take this embryo and peel off the skin like this, you can see these blocks of tissue that I showed you before. There's a block here, and here, and here, and here, and here. Okay, well, that's what they look like. There's one block right there, there's one block right there, there's another one there, and another one there. Those are called somites, okay, so remember that term. Okay. The lymphatic hearts, and every organism, every animal has a certain number of somites. A frog has 42 somites, okay, from head to tail, 42 somites. The lymphatic hearts form right between somites 3 and 4. Okay, so my working hypothesis is that the muscle cells for the lymphatic hearts come from somites 3 and 4. If that's true, then I should be able to remove somites 3 and 4 and put them in another animal, and they'll form lymphatic hearts, or I can put them in a dish, and they should form lymphatic hearts. Okay, so here's one experiment. Those are the somites. Here I've exposed them again. This is somite 3, this is somite 4. Okay, so you go with little tiny needles under a microscope, no coffee that day, and peel back the skin, and you cut out somites 3 and 4. Okay, again, these are about maybe about ½ millimeter long. Okay, so they're really tiny. Okay, about a 50th of an inch. Okay. And then you can put those in a dish. So if the muscle cells are coming from somites 3 and 4, you should see muscle cells form from that. If you put in somites 5 and 6, you should not see muscle cells. Okay, so it's conceptually, it's a pretty simple experiment. It turns out to be maybe not quite so easy to do, but so here's what happens. You put those somites in a dish, and some stuff grows out. Here's a little bit of skin that was contaminating. That's a little bit of skin. There were a few nerve cells around, these little spindly fiber-like things are nerve cells. And then there's this patch of tissue right here. And if you're sitting there looking at it under the microscope, it starts doing this. So this was wild when I saw this in the microscope, because most of the time the cells just sit there and they don't do anything that you can see. But these were twitching. This is approximately real time. So this is sort of the Frankenstein moment. Oh, it's alive. You know, so. All right. So this suggests that these somites really are giving rise to these lymphatic hearts cells. But that's not the end of the story, because it's a little difficult to get the these somites out clean every time. So there could be some other contaminating cells that are in the way. So I still have to work a little bit to clean this up, but it's on the right path. Okay, I have just a couple minutes left, which is good because this is the last slide. And I've made a little less progress on this one, but in some ways this is the most interesting project that's going on in the lab, and that's this. How are the lymphatic hearts counted and specified? Okay, so remember, there are, in this tadpole so far, I've told you that there are 2 anteriorly up here in the shoulders, and later on in development there'll be another 3 or 4 down here. Okay? But it turns out that if you look at other species, they have different numbers of lymphatic hearts. Okay? So the question is, it's sort of like, why do we only have 2 arms and 2 legs? Why aren't there some animals that have, you know, multiple arms or multiple legs? Well, it's hard to do that experiment because all the vertebrates we know have just 2 arms and 2 legs. But here's an animal that in some species it has 2 hearts, and in some it has 8 hearts, and in some it has 6 hearts. In some it can have as many as 200 hearts, all the way down the body axis. So here's a salamander, and it has lymphatic hearts up here where the forelimbs are, and back here where the hind limbs are, and then at every body segment in between. Okay. And here's what this animal looks like at a higher magnification. There's a heart there, there's a heart there, there's a heart there. Okay, so animals can make them anywhere, but frogs only make them in some places. And so the number and the location is under embryonic control. They must have some sort of signals to make them. That is, a frog can make, we know that animals can make them anywhere. Frogs make them here and don't make them there, but we know frogs can make them, so they must be turning off, frogs must be turning off the genes in here that are turned on in a salamander or some other species. So, so that's the future of the project, is to try to work on this and try to come up with some clever ways to figure out how the animal puts them in the right places and counts the right number.